my cutest little champions. I hope that you're doing wonderful, and from my end I want to wish you all a very happy new year. I hope this year brings the best out of you, everything good reaches you, and may you achieve everything nice, whatever you wish for, may you achieve that, may you get lots and lots of love. Anyways, I am giving you lots of love. Apart from me also, there might be people around you who are wanting to give you lots of love, so you will be receiving that possibly this year. Yes, and wishing you good luck and everything nice. Okay, and I hope that you have taken New Year resolution. It's fun to take New Year resolution, and I hope it's also fun to follow it. I have taken mine. I'll tell you towards the end. If you have taken yours, you can tell me if you want to in the comment section, and check whether you are following it or not. Chalo. First things first. So in this particular session today, we are going to decode the chapter that is molecular basis of inheritance, which is a very important chapter, carries 13 marks, right? So I will be telling you what you should not be missing in this particular chapter if you are someone who is revising the chapter, or probably you are studying for the first time. So these things you should not be definitely missing. Okay, I'll be telling you certain questions also, or what type of questions could be coming from this particular paragraphs. Okay, so let's begin. The color tone might change because today our editor is on leave, New Year leave, so probably adjust for some time, but I hope everything that is here is visible to you clearly. Yeah? Okay, chalo. So DNA, my dear children, over here, this statement that DNA is a long polymer of what? Deoxynucleotides. So you should be knowing deoxyribonucleotides. You should be knowing what is the monomer of DNA. What is it made up of? What is the simplest unit the DNA is made up of? Also, the haploid content of human DNA is 3.3 into 10 to the power 9 base pair. This is very much important. Haploid means half the number. It is haploid content of DNA is 3.3 into 10 to the power 9. This question is asked in the format of this, like number of nuclear pairs present in the DNA of primary oocyte of newborn in human. So everywhere we have diploid number of DNA, but only in the gametes we have half the number of DNA. Okay, or half the amount of DNA, not the number of DNA, half the amount of DNA. So here primary oocyte is being mentioned. That means you're talking about gametes, and gametes, as I said, it will be having half the number of half the amount of DNA. So it will be 3.3 into 10 to the power 9 base pairs. So you should be knowing these small things. This is how questions could be framed. Okay, now going ahead, this is very much important, the structure of the polynucleotide chain. They have mentioned that basically nitrogenous base is there, pentose sugar is there, and a phosphate group is there. So three things makes up the DNA. You should be knowing that. Okay, you should be knowing that. And also one more very important thing is given, that is uracil and thymine is there. In the case of RNA, uracil is present. Okay, instead of what, children? Instead of thymine. So there's a question that have come which is like this: pyrimidine base pair present in RNA in place of thymine of DNA is. So you know, in place of thymine, what do you have? You have uracil. And uracil, thymine, cytosine, all of them, they are known as pyrimidines. All of them, basically, they are known as pyrimidines. So nitrogenous basis, the DNA is made up of three things, right? Or basically the nucleotide. When I say DNA is made up of nucleotide, I say nucleotide is made up of three things. What is that? What are those three things? Nitrogenous base, pentose sugar, and phosphate group. Okay, now I am saying nitrogenous bases can be of different types. Either it can be purine or it can be pyrimidine. In purine you have adenine and guanine, and pyrimidine you have cytosine, uracil, and thymine. In the case of RNA you just have uracil and cytosine. In the case of DNA you have thymine and cytosine but not uracil. So you should be knowing it. And what are what? Okay, pyrimidine, purine, everything. Now going ahead, what do we have over here? They have mentioned what is there extra in the case of RNA. So in RNA, every nucleotide residue has an additional OH group present at second position of the ribose. So in the sugar that is there, ribose sugar, which is common to RNA and DNA. I told you there is one sugar, pentose sugar, right? That is ribose sugar. So in the second position of the ribose sugar of RNA, it has an OH group. And this OH group, I am telling you, makes the RNA molecule really unstable because it is highly reactive. Okay, so I just listed you one thing. So now, the people who were behind the camera, so Friedrich Mischer is there. He was the one who basically said that there is a substance present in the nucleus, acidic substance. That is what? Nuclein. He called it nuclein. And he didn't call it DNA, he called it nuclein. Then James Watson and Francis Crick came forward, and based on the X-ray diffraction produced by Maurice Wilkins and Rosalind Franklin. So these are the two people who did the X-ray diffraction. And on the basis of that, James Watson and Francis Crick gave the double helix model. So you should be knowing who gave the double helix model, my dear children. Now there's a very important question that comes here. what are the salient features of double helical structure of DNA? So you should be knowing all these points that is mentioned over here. It is this, the polarity is 5 to 3, the other one is 3 to 5, everything. How many hydrogen bonds are there between cytosine and thymine, how many bonds are there between guanine and not cytosine and thymine, adenine and thymine, and also guanine and cytosine. Okay, so basically there are 2 hydrogen bonds between adenine and thymine and 3 hydrogen bonds between guanine and cytosine. So you should be knowing about this. Now 2 chains, what is the distance between each base pair? You should be knowing about that also. So it is basically the pitch of the helix is 3.4 nm, that nanometer, you should be knowing that. Okay, and there are roughly 10 base pairs in each helix. So this pitch, okay, just a second. Everything that I am touching is okay. So over here you see this whole thing is a helix. This whole thing that you see is a helix. So how many base pairs are there? 10 base pairs are there in one particular helix. Okay, you should be knowing that. Okay, and in between that, what is the space? So there is a space of 0.34 nanometer. All this small information are really important for you to remember. Okay, then apart from that, okay, who gave the central dogma? What is the central dogma says? That is DNA will replicate itself, DNA will undergo transcription to form mRNA, mRNA will undergo translation to form proteins. This is the central dogma which was proposed by Francis Crick, my dear children, the same guy who gave the who said that there is a DNA double helical structure. Okay, please remember that. No. Okay. Over here again, very important piece of information. If the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pairs with distance between 2 consecutive base pairs. So this is the total number of base pair, that is 6.6 into 10 to the power 9. Okay, and then if you calculate the distance between each base pair we just studied, it is 0.34 nanometer, right? So it comes out to be 2.2 meters. That is the whole length of DNA. It's very much important. So how do you take care of this big DNA? You have to put it inside the nucleus, right? How do you manage it? For that, we have something that is known as nucleosome, my dear children. So they have mentioned about the structure of nucleosome. So you should be knowing that nucleosome is made up of what? Histones. Histone are what, children? Positively charged proteins. Histone is positively charged protein. Remember, DNA is negatively charged. So negative thing comes and coils around the positive thing, that is histone. Now histone comes in octamer, that means 8 histone molecules are there. Histone, why is it positive? Because it is a protein, so it is made up of amino acid. But what amino acids are more in number, which makes it really positive? There are different type of amino acids. Negatively charged amino acids are also there. So you should be knowing the name. It is lysine and arginine. It is mentioned over here. It is lysine and arginine which makes the histone protein, what, children? Positive. Now over here, as I said, the DNA wraps around the histone octamer, okay, and almost 200 base pairs of DNA is being there, okay, in that particular helix. So it wraps around twice. So for example, this is the octamer, it will wrap around twice. Okay, please remember it. And now the nucleosome. So basically I had the whole DNA, now I am trying to make it compact. Right, so this nucleosome and then my DNA, another nucleosome and then the DNA. This whole structure that you see is known as chromatin, and chromatin is also known as bead on a string like structure, because you will be finding what's a histone proteins like beads, and it's like a string. A string is what? Basically the DNA. So again the the chromatin, no, it can be categorized into 2. One is euchromatin and the other one is heterochromatin. Very important question. They ask you the difference between euchromatin and heterochromatin. So basically, when the nucleosomes are kind of apart, that means the chromatin is still there. Okay, that's why chromatin basically means you have the nucleosome and the DNA, right? Suppose this chromatin is condensed, more condensed. Now the nucleosomes would be close. Now the 2 different nucleosomes, it will be close. This structure you call it as heterochromatin. Okay, when they are loose, when they are far away from each other, a bit far away, you call this as euchromatin. So there are 2 types of chromatin fibers. One is euchromatin, and if they are close, you call it heterochromatin. So if it is euchromatin, okay, if it is open, it becomes easy for transcription to happen. So we say euchromatin is said to be transcriptionally active chromatin. Heterochromatin, basically they are close. Okay, so when you want to have a very compact structure of the DNA, that's when you have the chromatin, and the chromatin again becomes compact to form the chromosome. Okay, to form the chromosome, that is the most condensed form. Now in this most condensed form, I will call this chromatin as heterochromatin, my dear children, and it is transcriptionally inactive. Please remember that. So there's a difference that is being asked. You have to frame your answer. Now, all the questions that I am telling you over here, I have explained everything in the PU2 Brahmastra series. Okay, if you go, just check the second video of PU2 Brahmastra series in biology. It is molecular basis of inheritance, my dear children. I have solved all the questions. I told you how you should be framing your answers. So, watch that video. It is very, 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 very helpful for you. Okay, it will be worth your 2 hours or basically 1 hour, 30 minutes, whatever it is. Now, what is basically the genetic material? Okay, it was DNA, but it was not proved really early. Okay, there were so many experiments around it. So there was transforming principle experiment by Frederick Griffith. They asked you to explain the whole experiment, so you have to know it. Okay, what was the thing that was being used? It was Streptococcus pneumonia, my dear children. It was something that was causing pneumonia, and there were 2 strains. One is S strain and the other one is what? R strain. So the thing that I've highlighted over here is something that you have to put your attention on. Okay, all the experiments you have to study anyways. Okay, there is no way that you can skip it anyways, you have to read the whole thing. But these information that I am telling you, it's very, very important. They can ask you, what was the organism on which Frederick Griffith worked? So you should be knowing it is Streptococcus pneumoniae. And what was the strain? It is S strain and R strain. And since there are so many experiments, you will be confused also. So better you write it and it will be all the more clear. So this is one experiment that you have to study. Still, it was not confirmed that DNA is genetic material. Okay, so another guy came in, that is Avery, Colin, MacLeod and McCarthy. They all came in. Okay, they did the other experiment where they used what? Certain enzymes. They used protein digesting enzyme, they used RNA digesting enzyme, they used DNA digesting enzyme, and they found that when DNA digesting enzyme was used, the transformation was not taking place. So that's how they said that it's probably the DNA that is the genetic material. But still it was not clear. So then came the other guys, that is Alfred Hershey and Martha Chase, who were the final people who said that finally, yes, DNA is the genetic material. So again, what did they use, my dear children? They used bacteriophage. Bacteriophage is a virus. It is going to infect the bacteria. And what is the bacteria that is going to infect? Escherichia coli. Please remember, it is Escherichia coli. You have radioactive phosphorus and radioactive sulfur as well in this particular experiment. E. coli bacteria was used. Please remember that. Everything else is very important. Go through it. Okay, read it. And what were the steps? Infection, then blending, then centrifugation. Now, properties of genetic material, RNA versus DNA, what is important? The question comes, mention the criteria for a molecule that acts as a genetic material. So you should be knowing what all should be the features of a genetic material. So it should be able to generate its replica, it should be chemically and structurally stable. Okay, it should be having a scope for slow changes so that evolution can happen, right? And then it should be also able to express itself in the form of Mendelian characters. So these are the features, these are the features that you should not be missing while studying. Okay, this is how the questions also get framed. Just give me a second. My hair is causing trouble. Okay, cool, works. Apart from it, what do we have? Okay, this is a very important thing which explains that why RNA is not a good genetic material. Why it was, okay, the first genetic material that was identified was RNA. Okay, initially organisms had RNA as the genetic material, but it was kind of risky to have it. Why? Because RNA is something that could undergo mutations really, really fast. And if our genetic material is undergoing mutation, probably we won't be looking the same. We would be having so many changes, we would be having so many diseases. That is why the shift was made to DNA, which was more stable. So why it is not stable, that is RNA? 2OH group present at every nucleotide in RNA is reactive group and makes RNA labile and easily degradable. RNA is also known to be catalytic. That means it helps in processes to happen. It acts as an enzyme. Okay, hence they are reactive. So basically we should not be opting for something reactive, you should be opting for something stable, and that is stable. That stable thing is what? DNA. In fact, the presence of thymine at the place of uracil also confers additional stability to DNA. So as I said, DNA has thymine, not have uracil. So again, it gives additional stability. So you should be knowing all this two things, very, very important. RNA world, RNA was the first genetic material. As I said, RNA used to act as a genetic material as well as a catalyst. Okay, it's very much important. But then RNA being a catalyst was reactive and hence unstable and was later replaced by DNA. Now replication, Watson and Crick, the same guys, double helix model. Okay, double helix model. So they said that, okay, when DNA replication is happening, one strand is parental strand, another strand is new strand. So am I not conserving one strand? Yes. So I call this method as semi-conservative mode of DNA replication. But these guys said it, okay, but someone had to prove it. So I'll tell you about that guy, but let's see the question first. DNA replication is said to be semi-conservative. Why? So you should be knowing why is it semi-conservative? Because one strand is parental and the other one is newly synthesized. So the parental strand, am I not conserving it? Yes, which is why it is semi-conservative. I'm not using the entire strand. I'm just taking one strand of the parent. Right, so it is why it is semi-conservative. So describe the experimental proof of Michelson and Stahl to show DNA replication is semi-conservative. That is given over here. This is Michelson and Stahl experiment. Again, what was the organism that was being used? Could be asked. It is Escherichia coli. Okay, it is Escherichia coli or E. coli, and over here they have mentioned the medium in which everything was done. Go through this particular thing, very, very important. As I said, if you're finding a difficulty in understanding the concepts, please watch the video, Brahmastra series video. It will be really, really helpful for you. I am guaranteeing that. Okay, semi-conservative mode of DNA replication was demonstrated by Matthew Michelson and Franklin Stahl. Again, a question. These type of questions could be coming in. Okay, which is why I put the questions over here. Children, I hope this whole thing is helpful for you, right? I don't think it's just, please don't feel I'm just blabbering things out. Okay, I'm going on fast. I'm going fast because otherwise you'll get bored. I just want to tell you what is important. I'm not explaining the concept and I don't want to waste any bit of your time. So I hope you're understanding things. If you think that this video is helpful, do let us know in the comment section. We'll be really happy. Now, experiment was done on Vishya faba also, which is faba beans, okay, by Taylor and colleagues in 1958. By any chance, if it is asked, you can say. The same semi-conservative mode of replication, it is there to prove that one experiment was done in Vishya faba also by Taylor and colleagues. This is a whole different set of guys. Now, what do we have? We have DNA replication, my dear children. Okay, we were talking about that. So you should be knowing DNA replication is done by DNA dependent DNA polymerase. What dependent? They are dependent on DNA only. Okay, so DNA dependent DNA polymerase does it. Okay, and human whose diploid content is 6.6 into 10 to the power 9 base pair. So we saw the haploid content. Half content was 3.3 into 10 to the power 9 base pair. So diploid content, that is double. Okay, so it is 6.6 into 10 to the power 9 base pair. All our cells have this. Only gametes have half the number of DNA. Half the amount of DNA. Okay, now they have mentioned about DNA cannot be separated in its entire length. Why? Due to high energy requirements. Okay, there are high energy requirements which could be breaking down the hydrogen bonds, which is why it cannot be separated in the entire length. It is separated, like for example, replication is happening over here. So DNA separation would be happening over here. Others would be still joined. So this fork sort of thing that is being formed is known as replication fork, my dear children. Very important piece of information. DNA dependent DNA polymerase catalyzes polymerization only in one direction, that is 5:3. This direction only. Now, on one strand, the template with polarity 3:5, the replication is continuous, while on the other strand, the opposite strand, it is discontinuous, my dear children. Okay, who are the ones, which is the enzyme that is responsible for joining the DNA fragments? Later on, it is DNA ligase. Okay, so DNA, I will tell you three enzymes. Okay, one is DNA polymerase, which helps in the replication process, okay, which helps in making the copy. DNA helicase is there, which breaks the DNA, which makes it open so that the DNA polymerase can come on act. Okay, then DNA ligase is there, which ligates thing, which helps in the joining of things. It's actually like a Fevikwik. Okay, you can remember like that. Ligates. Now, DNA polymerase on their own cannot initiate the process of replication. Please remember that. Okay, it needs a definite region, and this definite region is known as origin of replication, my dear children. Origin of replication. No. The replication of DNA takes place in which phase? S phase. Okay, synthesis phase of the cell cycle. So the cell cycle is divided into different phases: G1, S phase, G2, and then the M phase. So S phase is the one where DNA replication happens. Okay, now this is the picture. Please remember it. In one strand, as I said, the one with polarity 3:5, it is continuous. Okay, so this is known as continuous strand. Over here, this is discontinuous, because as I said, DNA polymerases acts in what? 5:3 direction. 5:3 direction. So anyways, it has to come here, then again here, then again here. It's not a smooth transition. So it is discontinuous. Now transcription, I hope you know that. Now this is something that is not being asked, but I am telling you it could be asked, which is why I mentioned in blue. Why both the strands are not copied during transcription? So transcription is where only one strand is being copied. Why do you think both the strands are not being copied? So the answer to this is found over here. Please go through this paragraph. It could be giving you questions someday. So first, if both strands act as a template, they would be coding for different sequences, and this will create a huge lot of problem. Again, if two strands are transcribed together, these two RNA molecules which are being formed, they can also form a coil around them. If they form a double helix structure, do you think they can undergo the next process, that is translation? No. So this is why it is important. Okay. Now over here, what is the transcription unit made up of? Okay, so transcription unit will be having a promoter, will be having a structural gene, and will be having a terminator region. Okay, so this is the transcription unit, my dear children. Now over here, who helps in transcription? It is DNA dependent RNA polymerase. Again, it has the same polarity, that is 5 to 3 direction. So it will be working in 5' to 3 direction. So the other strand on which it is working, it will be having 3' to 5 direction. Remember the opposite. Okay, so RNA polymerase, DNA polymerase, all of them are going to work in 5' to 3' direction. Remember that. Okay, now the strand that has the polarity 3:5 acts as a template strand. It is known as template strand. The other strand is known as coding strand, my dear children. Okay, so this is the unit. I told you transcription unit consists of a promoter, structural gene, and a terminator region. Okay, now where do you say the promoter? How do you say? It is in the 3' end of template strand. Do you say like that? No, you always give a reference to this. It is in the 5' end. Okay, it is present in the 5' end, promoter. And here also, similarly, terminator is also present in the 3' end. That's how you say of the transcription, 3' end of the transcription unit. So the promoter and terminator flank the structural gene. The promoter is said to be located towards 5' end of the structural gene. So you say 5' end of the structural gene. Even though this particular strand is going to get transcribed, you give a reference to the coding strand, which is why you say 5' end of the structural gene. Okay, similarly, terminator is towards 3' end of the structural gene. These questions could be asked, which is why I am telling you. Okay, so it is a DNA sequence that provides binding site for RNA polymerase. It is present in the promoter. Okay. Construct a complete transcription unit with promoter and terminator on the basis of the hypothetical template strand given below. There is a template strand that was given below. I have not mentioned it here. Write the RNA strand transcribed from the above transcription unit. So they told you to make something like this, and when you are asked to have a transcription unit, how do you explain that? How do you explain? You have to make a transcription unit. That means you have to say, you have to put some sequences. A, U, G, C, G, A, things like that. So this is a strand. Okay, now if you want to make a complementary RNA strand out of it, so what do you do? Wait. I did a mistake. How do you make it come back? Okay, so this is a DNA strand, my dear children. It can be A, T, G, C, G, C, whatever. This is a template strand. And suppose this is 3' to 5' direction. So in 5' to 3' direction, the RNA polymerase works and it will be making a new strand. This new strand will be having, instead of A will be pairing with U, my dear children, this time. This is what? RNA. Similarly, T will be binding with A, G will be with C, C will be with G, G with C, C with G. So this is the new RNA strand that will be formed. So this is the question. You have to make first a transcription unit like this, where you have to mention A, T, G, C, 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 whatever on one strand, that is template strand, opposite of it on this particular strand. But remember, only this strand is going to get transcribed, this one. So you have to make a complementary strand like this and show. I have mentioned it here only, but don't do like this, okay? Don't do like this, write it separately. Don't mention it like this in the exam. Cool? In the exam, make complementary strands like this. Pick one and make a transcription. Pick one and make an RNA strand out of it. That's all you have to do. Now, how do I? Just a second. Okay. Come on. Chalo. This we studied. Okay. Remember, in the case of human beings, or basically eukaryotes, we are monocistronic. What do you mean by that? Basically, there is one cistron, okay, one transcription, there is one area, sequences, okay, in which there are multiple genes, my dear children, okay. There is monocistronic, as in there is one cistron Fine. And in one cistron, you would be having multiple genes. So these genes are basically having some space in between, which area that does not code for anything. You call these things as introns, my dear children. And the area that codes, okay, you call them as exons. So over here, the DNA sequence coding for tRNA or RNA molecule also defined as gene. However, okay, the structural gene and transcription unit could be said to be monocistronic, mostly in eukaryotes, or polycistronic, mostly in prokaryotes. Always remember, in the case of prokaryotes, they have polycistronic genes. Polycistronic genes as in one cistron means one gene, another cistron means another gene. Okay, they have multiple cistrons like this. In eukaryotes, the monocistronic structural genes have interrupted coding sequences. The coding sequences in the eukaryotes are split. The coding sequence or express sequence are defined as exons, okay, and the one that comes in between is known as introns. Introns are not found in prokaryotes, please remember it. We are monocistronic. In one cistron, we have multiple genes. In between the multiple genes, we have introns. Okay, prokaryotes do not have this. Please remember it. Now, in bacteria, there are three major types of RNA. Okay. Messenger RNA, this RNA, rRNA, and all this thing, very much important. Over here, something more important is coming over here. Okay. How is RNA polymerase able to catalyze the three-step? So RNA polymerase should be initiating this, elongating this, and terminating the process of transcription, right? So remember that there is a factor that is known as initiation factor, okay, which when it binds with RNA polymerase, that's when it can initiate. RNA polymerase can initiate the process of transcription. Now, when there is another factor, that is termination factor, when it comes and binds to the RNA polymerase, that's when it can terminate the process of what? Say, transcription. Please remember the names: initiator factor, that initiation factor, that is this, and termination factor, that is this. Now, in bacteria, since mRNA does not require any processing to become active, also since transcription and translation takes place in the same compartment, I hope you know that they do not have a nucleus and things like that. So everything happens in the same location, that is in the cytoplasm itself. So just when transcription is going to end, okay, or much before the transcription is going to end, translation would be starting there. But in our case, it is different. Okay, translation would be happening in the cytoplasm. Now over here, in eukaryotes, you have additional complexities, as I said. Okay, first is RNA polymerase. They are only of different types. You have RNA polymerase 1 that transcribes what? rRNA. Please remember what all type of RNA it transcribes. Okay, RNA polymerase 3 is responsible transcription of tRNA, 5srRNA, snrRNA. Okay, not rRNA, RNAs. Okay, RNA polymerase 2 is responsible for basically this, that is hnRNA, RNA, and also mRNA. Very much important. So write a note on the role of sigma factor and rho factor. So this is the sigma factor, that is your initiation factor, and then rho factor is termination factor. So you should be knowing their role. Okay, only when it comes and binds, that's when things would happen. Now there is something that is known as splicing, very much important. The questions have come like this: what is the name of the process where introns are removed and exons are kept in a defined order? So basically, this process is known as splicing, my dear children. What do you call this process? You call this as splicing. Okay, now when I say that DNA is made, mRNA is made out of DNA, still it has lots of processing and things to be done, which is known as capping, tailing, and things like that, after which it will be moving out, okay, and undergoing the process of translation. If this is not done, translation would not be happening properly. Please remember that. Okay, which sequence of bases transcribed from DNA are found both in hnRNA and mRNA? So when DNA is going to make mRNA, it's not a direct process. First, DNA will make hnRNA. Okay, hnRNA will also contain the introns. Introns are being cut with the help of splicing, and now you call this as mRNA. So what is common between hnRNA and mRNA? Both of them will be having exons, because exons are not being removed. Introns are being removed, exons are not being removed. So please remember that. So exons are something that will be there in both. Now you have this thing, genetic code. So there are 64 codons, my dear children. Okay, and you have to remember this. Also, the salient features of a codon, how the question comes, they can give you something like this. Given below the sequence of nucleotides in a particular mRNA and amino acids coded by it. So they said AUG is there, methionine is there, UU is there, phenylalanine is there, UU is there, phenylalanine is there, GAG is there. So they have given you things like this. Now write the properties of genetic code that can be correlated from the above. So you have to tell. So AUG over here, you saw it is basically methionine. It is coding for methionine, and AUG is something that is the start codon. If AUG is not there, it won't allow translation to happen. So these are the things that you have to definitely remember. And also UUU is there. which is coding for phenylalanine. UUC is there, that is also coding for phenylalanine. That means there can be multiple amino acids coding for, multiple codons coding for the same amino acids. So you should be knowing that also. It is degenerate. So you will say this feature shows that it is degenerate. Okay. Now the code is nearly universal for all the organisms. Okay, that is there. AUG has dual function. Now over here you saw UAA is there, but no amino acid is there. That means termination has happened. Okay, that means basically in translation termination has happened. So you should be knowing that. So these codons are terminator codon and things like that. So on the basis of this, questions can be coming. No. Okay, there's a example of a mutation that is there, sickle cell anemia point mutations example. So just go through it. Okay. And also one more thing, insertion or deletion of one or two bases change the reading frame. What do you call this type of mutation as? Frame shift mutation or deletion mutation. You look for this in the principles of inheritance chapter, but for confusing you guys, they have kept it over here. So please remember, something like this is there. Okay. Now what do we have? tRNA. So tRNA, the structure is very much important. You should be knowing there's an anticodon loop, there's an area where the amino acid comes and attaches. You should be knowing this is the 5' end, this is the 3' end. On 3' end only the amino acids will come and attach. Okay, so this is known as amino acid acceptor end, this is known as anticodon loop, everything. Okay, there is something that is known as initiated tRNA. Initiated tRNA will always have what? Methionine. Will always have methionine, because the codon which is there, it is AUG. It is what? AUG codon. And what is the structure? It looks like clover leaf, but actually it is inverted L. Okay, it is actually inverted L, all things like that. Now translation, the first phase, what happens in there? Okay, or basically what do you mean by this? What is the process of activation of amino acids in the presence of ATP and its linkage to their cognate tRNA known as? It is known as amino acylation of tRNA, or you call it as charging of tRNA. Okay, so in the presence of ATP, one amino acid comes and binds over this. Okay, so this is an energy utilizing procedure. It is known as amino acylation of tRNA or charging of tRNA. Now over here again, ribosomes, how many units it contains? It contains 2 units. Okay, then what is the first step? When the small unit encounters an mRNA, the process of translation of mRNA to protein begins. That is the first step, so you should be knowing about that. Okay, the ribosomes also acts like a catalyst. The ribosome which is there, it is acting like a catalyst because it has enzymes like that. Okay, there is something that is known as untranslated region. Very important question. They ask you what is UTR. You should be knowing it. There is untranslated region. So in the mRNA, before the start codon and after the stop codon, you have certain regions which is not going to get translated. These are known as untranslated regions, my dear children, and they are present at both 5' end and at 3' end. They are required for efficient translation process. For translation process to happen smoothly, it is there. So please remember, UTR very, very important. Now all the terminator codons begin with the nucleotide off. If we have seen the terminator codon, my dear children, all of it, this is a question that comes over here. It's stop. All of them basically is made up of uracil. That is the first one. Okay, so you should be knowing that it is uracil. UAA, UAG, UGA, all of them, 3 of them are stop codons. You should be remembering their name. Very much important. Start codon is only one, that is AUG. Okay, now for finishing this process of what? Translation, also you should need a release factor, okay, which will be binding to the stop codon, terminating translation, because there is no tRNA that will come and bind there. Okay, now regulation, okay, very important. Again, I mentioned the levels where gene expression can be regulated. These are the levels in the case of eukaryotes. In the case of eukaryotes, it can be in the transcription level or processing level, transport of mRNA from nucleus to cytoplasm. In the case of prokaryotes, they do not have nucleus, so basically everything is going on there. Okay, so they do not have these many levels. Okay, we definitely have these many levels. Please remember all the levels. Okay, formation of primary transcript, then splicing. If splicing is not done, translation will happen? No, children. So again, you are basically regulating the expression of the gene, right? If translation is not happening, then obviously protein won't be made. So this is the way. If you are blocking any of the process, it is going to hinder in the gene regulation. Now, okay, in prokaryotes, they have mentioned about certain proteins which are there, certain regulatory proteins which are there, okay, that can be positively acting, that can be negatively acting. Okay, these proteins which are being made, regulatory proteins, you call them what? Regulatory proteins. These regulatory proteins can be going and binding on to certain regions which is known as operator. So you have studied about promoter, you studied about this thing, structural gene and terminator. So just as behind, just near to the promoter you have operator region. Now on this operator region, if some bad protein, okay, negatively negatively effective protein is coming and attaching, the process of translation would not be happening. Okay, so you should be knowing about that. Not translation, transcription. So it was lac operon, which was given by Francois Jacob and biochemist Jacques Monod. Please remember the names, it is very much important. They have mentioned about what all the genes do. In the structural gene of lac operon, you have ZYA. What is Z coding for? Beta galactosidase. What will happen if beta galactosidase is not there? You should be knowing about it. What will happen if the next gene, that is Y gene, which is coding for permease, if it is not there, what will happen? You should be knowing about each of the things. Okay, so galactosidase will not be synthesized in lac operon if mutation occurs in Z gene. In the Z gene. These are the type of questions. Is it true, false, what? Should be knowing about it. So this is the structure. Please remember the structure, very, 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 very important. Okay. Lactose is a substrate for enzyme beta galactosidase, and it regulates switching on and off of the operon. So lactose is what? It is basically going to act like an inducer. It is one that will say whether it's on or off. Okay, if lactose is there, it will be on. Okay, lac operon will start working. Now the repressor of operon is synthesized from the I gene. Okay, repressor protein, I said repressor protein is being made. Any protein is made from a gene, right? So what is this gene? I gene. The repressor protein binds to the operator region of the operon and prevents RNA polymerase from transcribing. If some guy is standing over there, obviously RNA polymerase cannot go and transcribe things, right? So that is the problem of this particular thing. Now in the presence of an inducer, that is lactose or basically allolactose. If lactose or allolactose is there, what will happen? This lactose will go and bind with the protein. Now this protein, is it free to go and bind to the operator? No. So if it is not going and binding to the operator, obviously RNA polymerase would be able to transcribe there. Okay, so basically the lactose is going to act like an inducer. Please remember that. And one more very important thing, regulation of lac operon by repressor is referred to as negative regulation. It is what? Negative regulation, because the repressor protein is there, and repressor protein is always making the lac operon in off stage, in an off mode. So this is a negative regulation. Okay, and when lactose comes in, it basically helps in turning it on. Now human genome project, very much important. Okay, they ask you for the goals of the human genome project directly. So list the goals of human genome project. You should be knowing it, why it is important and everything. So knowledge of this will help in preventing a lots of diseases. If you know the genetic code, if you know what proteins makes what, we would be able to identify and basically treat a lot of diseases. Okay, now all this thing, yak, back, and then what do we have? Salient features of the human genome, very much important. Okay, very, 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 very important. All the points that you see over here, all the points, without even leaving a single point, you have to study, my dear children. I am writing here so that you don't forget. Okay, I didn't color it all because it won't look good. Okay, all points are very much important. Salient features of human genome comes like that only. Okay, now over here, back and yak is there just for your information, know it. Okay, where is back and yak? Back is bacterial artificial chromosome and yak is yeast artificial chromosome. They are going to act like vectors. They are going to act like what? Vectors. Yes. Now what we have? DNA fingerprinting. Okay. DNA fingerprinting, again, very important topic. They will ask you, with the help of VNTR mechanism, can you tell how things work? So VNTR stands for variable number of tandem repeats, my dear children. So they will ask you the steps, step by step. So you have to say that first isolation of DNA is being made, then digestion is being done, then separation is being done, then identification is being done with the help of hybridization and things like that. That's when we get to know that basically, oh, okay, fine, this DNA matches with the culprit's DNA, and this guy should be the guy who should be inside the jail. So all the steps, again, as I am saying, okay, I have explained it in very much detail. If you go and watch the master series, it will be really, really helpful for you guys. So that's all, my dear children. So DNA polymorphism, just know that. Okay, what is it? And anything else? No, yeah, DNA fingerprinting, what is it made up of? Basically, what is it dependent upon? Not made up of, dependent upon. It is made up of, dependent upon the repetitive DNA. So DNA fingerprinting involves identifying differences in some specific regions in the DNA sequence called the repetitive DNA, because in this sequence a small stretch of DNA is repeated many times, and this is unique to all. So that's on the basis of which the DNA fingerprinting mechanism works in a whole. So these are the important topics, my dear children. Please not miss it. Okay. Anything apart from this? Okay, that's all. And also, after you're done with the chapter, go read the summary, or before starting the chapter you can read the summary. It will help you understand what is a link. Sometimes you forget the link, and in this book now they have mentioned things, lots of things here and there, so I know it might confuse you so much, so better you read the summary and then start. It will be helpful for you. And with that, we are completing our today's decode NCERT thing, okay, of this particular chapter, that is molecular basis of inheritance. I hope it was helpful. Still, if you have any doubt related to the concepts, you can ask me. Okay, I will be definitely helping you out. Go watch the video. As I said, I might have gone really fast over here, but it's for just making you understand what is important and what is not. Okay, so please prioritize this chapter very much. This is a lengthy chapter. If you watch that video, it will be really helpful for you. Okay, so that's all, my dear children. Wishing you all a happy new year once again, and bye-bye. Take care. Do not forget to subscribe to our little channel, that is Diksha Karnataka.